So this is our presentation for MS Pickup Data Science Challenge. The problem statement that we have selected is Tiger Grant Challenge. Tiger stands for tumor infiltrating lymphocytes in breast cancer. As you can see, breast cancer is quite dominating and Asia stands as the second largest continent with 34% of mortality rate across the globe. Also, it's quite prevalent across various states in India. So what actually is Tiger Grant Challenge? So Tiger Grant Challenge is an initiative aimed at advanced computing pathology through assessment of various histopathology slides. These slides consist of whole slide images and various patches of the samples collected. Uh, the type of cancer that we are treating in this challenge is HER2 positive and triple negative breast cancer, which is very hard to diagnose as compared to other three types of cancers. And here, TIL, which is tumor infiltrated lymphocyte, serves as a powerful biomarker. Now, biomarker is something that tells us the prevalence and also about prognosis and diagnosis of a particular disease. The goal of this challenge is to provide an accurate, efficient, and scalable solution using computer vision and artificial intelligence algorithms. Initially, this process is done using manual examination, but now we provide an alternative solution to it, which will aid in faster diagnosis and broader accessibility. So, currently manual examination took a lot of time, and there are less pathology and limited expertise. So, we propose a solution which uses AI-powered diagnostic support and objective and reproducible assessments, which will lead to improved patient outcomes. So, what exactly is TIL? So, tumor infiltrating lymphocytes are immune cells which we can also recognize as lymphocytes or plasma cells, which are present in the whole slide images of breast cancer stroma. So here, as you can see on the left side, we have a slide patch image, and on the right side, the yellow bounding box are the TILs. Now, TILs has a special significance in detecting breast cancer, as it has high prognostic value and its prediction and TIL score gives us important stats. The dataset provided by Tiger Challenge is divided into three parts. The first one is WSI ROI, second one is WSI TILs, and third one is WSI bulk. The bulk contains various whole slide images in TIF format and its corresponding annotations in XML format. The ROIs, WSI ROIs dataset consists of ROI and WSI level annotations of various cell areas and TILs areas. WSI TILs consist of tissue masks of the affected cancer area in whole slide images. So what exactly is a whole slide image? The whole slide image is the stained image that we get after staining a sample. Now, there are three images here. The first image is the whole slide image. The second image, the black border which you can see, is the affected area or affected tissue, and the border, so the area which is present inside it is actually affected. The third image is patches. We can obtain patches by magnifying into whole slide images and getting patches of 512 or 256 pixels each. WSI are of high resolution and very hard to hover over or process. So we divide it into patches for training. Thus, there are mainly three tasks in the Tiger challenge. The first one is detection of lymphocytes that we saw earlier. Second one is the segmentation of the invasive tumor, which is segmenting the tumor. The segmentation task in this challenge is quite challenging, as the slides that we have got are stained slides and which have various color variations due to biological, chemical, and physical processes. The third task in this is computing an automated TIL score, which will actually give us the prognosis of breast cancer in a given slide. So let us deep into our approach. The first approach is of detection of lymphocytes. Here we have resized all the images into 512 pixels after applying pre-processing steps and converting them into PNG. Then using the JSON that we have got, we have applied the annotations over all the images. Here you can see that we have all the images in the yellow bounding box, which are actually the lymphocytes present inside the slide. We have applied YOLO-V8 detection model for training and inference. So this is YOLO-V8 is state-of-the-art detection model, which is quite accurate in real-life cases and in medical images. It provides a balance of speed and precision for detecting lymphocytes. Second approach is of segmentation. In the given image, you can see the ones there are TILs in across the image. However, we have the affected area which are called stromal TILs and not affected area. So we need to segment it out from the rest. In this, we have applied image processing and stain normalization. We have also trained a unit model in which we had 7 labels, including invasive tumor, tumor associated stroma, in situ tumor, healthy glands, necrosis, inflamed stroma, and rest. Here we have obtained the mask image after patching, and then we have trained a unit model to get the inference. Unit is basically used for segmentation in medical imaging. It is an encoder and decoder U-shaped architecture which keeps collections and allows to capture high-level context and fine details for accurate tissue segmentation. The third approach is computation of automated till store. Here we initially contain the tumor infected lymphocytes area, then total till area, and then we compute using segmentation and measure the stromal area. Then we compute the till score for prognosis, and at the end we obtain a till score for all the given slides. We compare it to find the diagnosis. So these are the results of our detection model. These are the accuracy metrics, and this is the confusion metrics. We have got about 79% of accuracy and 0.79% precision of MAP. This is the result of our segmentation model, where we have segmented all the 7 layers as discussed earlier. This is our walkthrough of the approaches, where these are the tasks, these are the approaches as described earlier. This is our code for converting whole slide images into patches. This is our training script for training the Euro model for description. These are the various parameters that we have set to get the best accuracy. We used two models, nano and medium, and we finalized the model which had the highest accuracy. Here we have performed inference over it. This is the training and processing code for segmentation model, where we have applied various pre-processing steps and did color and stain normalization for histopathology slides. Here we have formed a network architecture and did training on around 70 epochs. We have considered Jaccard loss, which formula is given, and we got some good results with it. Later, finally, we applied inference over the images, and these are the inferences we are getting. One special thing which we did in this process was to patch and unpatch the image. As we know that in medical images, it is very crucial to get all the features. And when we resize or demagnify an image, we might be losing some important factors. So what we did whenever we got in whole slide images, we divided into the patches and then gave it into the training and inference model. And at the end, we unpatched the whole image, unpatch the whole patches so that we do not get any loss of data. This was the computation of till score and results as we discussed earlier. This was our project by Viren Mehta, Perin Modi, and Tirth Patel. Thank you.